My name is Matthew Leming. I'm a senior computer science major here at UNC. My research mentor is Dr. Martin Styner, that's back over there. And what I'll be talking about today is a framework for incorporating Diffusion Tensor Imaging Atlas Builder registration into tract-based spatial statistics and a simulated comparison of that to standard tract-based spatial statistics. Okay, so I'm sorry for the word salad right there, but I'm going to try to provide some background. Okay, so what I will talk about today, first I will begin by going into a background. What is diffusion tensor imaging? What is tract-based spatial statistics? How am I attempting to improve these things? I will then go into the use of directional data in tract-based spatial statistics and sort of my work in that area. I will then go into the use of single value measurements of diffusion tensor imaging in tract-based spatial statistics. And then finally, I'll go into the ways in which I'm testing out my sort of improvements to these things to the original version of TBSS. So the first thing I'm going to talk about, I suppose, is diffusion tensor imaging and what it is. Okay, so basically, diffusion tensor imaging is a form of MRI, magnetic resonance imaging, which is used to find information about white matter in a person's brain. Now, everybody here probably has heard about white matter and gray matter in brains. The difference between white matter and gray matter is that gray matter is essentially clusters of neurons where a lot of computation is carried out, and that's basically where thinking and processing is done. And white matter is sections of parallel neural fibers or tracts, which are used to connect these areas of gray matter. And diffusion tensor imaging is useful because while it doesn't give that much information about gray matter, it can model systems of white matter in a person's brain. Okay, and it does this, and the way that it ends up modeling these things is through a number of what is called tensors. I've drawn an example of a tensor on this whiteboard. I don't know if it's the best drawn one in the world, but essentially a tensor is a kind of a 3D ellipsoid which can indicate a particular direction in which a white matter tract is pointing, and so this here is just a picture that I pulled that can kind of show how a number of these tensors can end up representing information about the image of a brain. So you can sort of see here in the middle, in the middle of the brain, there's a bunch of spheres. That's because those are ventricles. Basically they're full of fluid, so they don't really have much of a direction. And you could sort of, you could see that they're a lot less kind of directed than say, the areas right here that are visualized, which are very straight and kind of tensors that indicate the presence of white matter. Okay. All right, and so within DTI, within each 3D pixel of data, you have one of these tensors that consists of 6 numbers, which can represent the tensor. So you have three directions that you need to indicate. You need to use to indicate a particular direction, white space, in 3D space, and you also have the 3 sort of radii of each of these tensors, which is indicated by the first eigenvalue, which you can see right there is represented by lambda 1, and the second and third eigenvalues as well. Okay, and so that's what DTI is, and however, when you're trying to actually visualize these sorts of things, it's often difficult to work with totally 3D tensors in each voxel, and so very often people will come up with one-dimensional measurements of diffusion of DTI, and one of the most popular ones to use is what's called fractional anisotropy. Okay, And I don't actually, unfortunately, I neglected to write fractional anisotropy right here, but that's just FA, okay, which is indicated by this equation. Essentially what you need to know about FA is that it measures sort of how directed each one of these tensors is. So a sphere, so it's a value between 1 and 0. A sphere would have no direction, therefore it would be indicated by an FA value of 0, whereas a very sort of directed sphere would indicate a very high FA value, close to 1, and an FA value close to 1 would indicate would say that there's probably going to be white matter in this area, and it's probably going to be, you know, very, very highly directed. Okay. And then these are just other measurements of DTI, and they're a lot simpler than fractional anisotropy. So you have radial diffusivity, which kind of measures is a measurement of the thickness, I guess, of the tensor. You know, it's the second and third eigenvalues. The mean diffusivity, which is just the average of all three, and then the axial diffusivity, which is the largest eigenvalue in the tensor. Okay, so that's just explaining what these data formats are. You know, these are images of people's brains, and so okay, how do we use diffusion tensor imaging? What is its purpose? Okay, so this is where TBSS or tract-based spatial statistics comes in. Now, basically, the purpose of TBSS is if you have like 50 diffusion tensor images of, you know, people who are control patients, you know, perfectly normal people, and then 50 other images of people with, say, autism, or who have been abusing a certain drug, or who have some reason to believe that there is going to be a difference in their neuroanatomy. We just basically put all those images together, we run them through TBSS, and then TBSS can say, okay, these are the areas of white matter that in which these people vary. And we can therefore, and then we can kind of assess from that, that is suggestive that any one of these disorders affects white matter in that particular area. Okay. And so it's very useful because it's kind of automatic sort of push of a button thing. It's very easy to use by neurologists and psychiatrists that would be performing these studies. And so that's one of the reasons that it's very widely used, and it's kind of gotten very popular in the last since it was released about 10 years ago. Okay, and so this is sort of the process that TBSS goes through. So the first thing it does is it pre-processes everything. So it takes all the diffusion tensor images, and it takes all those and it estimates the FA, the fractional anisotropy images from them. So you'll have a bunch of images that are very kind of bright, where there is white matter tracks, and essentially where the diffusion is high, there's going to be a higher intensity, closer to 1. Okay, so the first step is just pre-processing. You do that, you know, you're good. The second step is called registration. So essentially, all of these images are deformed so that similar anatomical structures are in the same space. So if with unregistered images, if you lay them on top of one another, you know, the brainstem of one person is going to be like over here. The brainstem of another person is maybe in a totally different area or just a few voxels off. And so you can't really do a whole lot of group comparison studies with unregistered images. So basically, this takes the images and kind of deforms them so that if you take a certain coordinate of each image, you can be assured, you know, this is this particular area of the corpus callosum, it's right here. Or if I take a voxel here, it's going to be the same area of the brainstem for all the patients. And so that's the second step. The third step is where a white matter skeleton is estimated for the entire brain. So you'll basically have kind of this this mask over the entire brain that's covering kind of all the major white matter regions in the brain. And I'm just going to skip a slide just to show you an example of that. And so you can see this is kind of an average of all the images in a particular study, and this is sort of an estimate of where the white matter is in this particular study. Okay. And after that, what it will end up doing is it will kind of go through all subjects and then do a search in areas perpendicular to this skeleton, and it will end up projecting the highest fractional anisotropy value it finds in a localized region onto the skeleton. Okay, and then what you have is basically a bunch of numbers which you can compare across groups to find average differences in this FA across two different groups of people. And so this particular visualization, which I pulled from the TBSS website, kind of shows areas where it detected statistically significant differences in FA between these two groups of people. Okay. Now, okay, so yeah, TBSS, it's a tool which can tell you kind of the average differences between two groups of people just based on the images of their brain. Now, the problem with TBSS is that it basically takes all this information about DTI, which has, again, 6 components per voxel, and it pretty much just throws all of it out in the first step. Okay, and that's done because it's a lot easier to work with just single-valued FA than anything else, than a 3D tensor. But it also carries with it some problems. So a piece of white matter running front to back, as well as an area of white matter running top to bottom. They could end up having the same fractional anisotropy value, and so what you're dealing with in that case is could be TBSS saying, okay, these are totally indicate the same area of matter of white matter, whereas it's just not true. So yeah, at the end of the day, TBSS just ends up throwing out a lot of data that ends up causing a lot of problems. And yes, okay. So what I'm trying to do is find a way to kind of use that data that is kind of thrown out by TBSS at the very beginning. So the way that I would end up doing this is by kind of finding out how to incorporate this data into each step. Okay, the first and probably one of the most obvious ways to do this is to incorporate that into the registration step. So there are already kind of registration algorithms which TBSS in its current form just doesn't happen to use, which incorporate this directional data when you're taking each subject and you're putting it into the same space. So basically, you take an algorithm which doesn't just estimate the FA of all subjects and then it just puts them into the same space. It actually tries to take the tensors and put those into the same space, which is very useful. After that later steps of TBSS, there's been a lot less research in actually incorporating any kind of directional data into this, and so that's kind of an open area of study. What I tried to do was sort of use other measurements of diffusion tensor imaging, such as, some are written on the board over there, axial diffusivity, medial diffusivity, and radial diffusivity, and attempt to compare those instead of just doing it with FA. So, it's definitely not a perfect solution, but it also hasn't particularly been explored before by people who do study TBSS. So that's what I tried to do. And okay, so this project kind of culminated into something called DAB-TBSS, which is DTI Atlas Builder, Tract-Based Spatial Statistics. Essentially, I took an algorithm called the ANTS-SYN algorithm, so Advanced Normalization Tools Symmetric Normalization algorithm, and I used that to perform the registration step in TBSS. And then in the later steps, I attempted to compare not just fractional anisotropy, but also axial diffusivity when I was performing group-based comparisons. Finally, one of the kind of more creative aspects of this project was actually finding a way to test which is more effective, the original TBSS or my alterations to it. And I did this by coming up with something that actually simulated differences in each of the data sets. Okay, and so basically this ended up And this is just the method used to compare the two methodologies, which ended up being something called synthetic DTI differences. And this is essentially a MATLAB script, which takes, in which you can give it 20. I give it 20 subjects, and it can go in, and I select kind of a sphere-shaped space for each subject, and then what it ends up doing is it increases the principal eigenvalue by 5, 10, 20%. And so what you end up doing is you take these subjects that have simulated differences, and you take the same number of subjects that have that are just completely normal, and you end up comparing them. And basically, if TBSS or my version ends up actually finding these differences, so saying, I see what you did, Matt. There's a difference in these three areas. Then you say, okay, this is correct. This did its job. Likewise, if it ends up coming up with false positives in other areas, then you could say, okay, well, I know there's no differences there, so you're wrong. Okay. Okay. So, and this is just kind of a visualization of what it's actually doing. So the simulation just kind of takes each tensor and just in the cases I did, it literally kind of extends each one by a certain percentage. Okay. So, yes. Okay. So the results. So the specific tests that I ran, I took 20 subjects from another study that I was doing. I kind of put. I indicated for each subject the ventral corpus callosum, the middle corpus callosum, and the dorsal corpus callosum, and then I ended up in each of those in a kind of a sphere-shaped area. In all of those subjects, I increased the principal eigenvalue by 0.05%, 1%, 2%, 5%, 10%, and 15%. So just coming up with a lot of differences and intensities, and then seeing how intense, how large do these differences have to be before TBSS can actually end up tracking or indicating those differences. Okay, and the conclusion I found was that by and large the DAB-TBSS, so my version, when it was measuring specifically axial diffusivity, so not FA, was the most successful in this particular case at detecting differences in those three areas. So so what happened when I just ran straight at fractional anisotropy. Actually, in this case, TBSS actually won, but even so, I had to go to pretty great increases in the magnitude of the principal eigenvalue before it actually detected any of those differences, and even then, it only detected 2 out of 3. So you can see right here. This is the result using regular TBSS, which it managed to detect statistically significant differences there, but not in the dorsal corpus callosum. And then mine didn't actually detect the differences. Like DAB-TBSS didn't actually detect differences, period, when it was just measuring fractional anisotropy. And again, this is after between groups the average principal eigenvalue was increased by 15%, which is actually a pretty huge difference. However, when we're just studying axial diffusivity, which is just testing directly the magnitude of the principal eigenvalue of the tensor, what happened was actually pretty weird. So DAB-TBSS detected all 3 differences, and these are both visualizations of DAB-TBSS. You can see the yellow voxels are where it detected the differences. It successfully detected them in all 3 areas without any noise. And TBSS actually did do detection at much lower rates, but it also came up with a number of false positives, like just all across the board when we were testing for these things. So, I didn't actually have the visualizations right here, but so so regular TBSS was saying, okay, so I see a difference here, here, which is just, I wasn't even simulated, period. So that would basically tell if a physician were using that, they would end up coming up with a completely wrong conclusion. Okay. So in this case, the conclusion is that TBSS, basically TBSS did find differences in 2 out of 3 areas for 15% change in magnitude, but for a smaller change in magnitude, DAB-TBSS was more successful at detecting these differences upon only a 10% increase in the principal eigenvalue, and because 10% is lower than 15%, so it detected a more solid difference successfully, DAB-TBSS wins. Yes. So kind of the future work of this, of course, is trying this out with more subjects, so increase the number of subjects to 30 or 40 or 50, and finding out, how many subjects are necessary so that it can detect, it can successfully detect much lower differences between them. Another kind of obvious route to go, and also the reason I didn't do this right here is just because the time it takes to actually compute these is pretty lengthy. So it takes like several days on the kill double cluster to run 20 subjects with TBSS, and it takes exponentially more time to run it with 30 or 40. But obviously that's something I could do over the summer. So another obvious kind of thing is to vary the second and third eigenvalues, and then also test for variations in radial diffusivity or medial diffusivity. In this case, I mainly was interested in axial diffusivity because that is directly what I was varying in this particular case. I was also interested in fractional anisotropy because it's the most commonly kind of used value in this field. So, yes, that 2135. All right. So, any questions so far? That's about it.
Question?
Oh.
Do you have an explanation for why DAB-TBSS can distinguish finer differences but not the larger ones?
I'm actually that's going back to this slide. So the reason I would guess so first off with finer so with regards to kind of the finer differences, I think it would be able, just with the way these work, it would be able to detect finer differences if you kind of threw in more subjects and it said, okay, I can determine just kind of based on the fact that I can see these differences on average in 30 subjects that these differences are present. I think that sort of the reason that TBSS in this case with fractional anisotropy was able to do it was because it's sort of working directly with FA values, whereas DAB-TBSS is working directly with tensors. So basically, it's sort of performing deformations directly on kind of higher values of fractional anisotropy, whereas that wasn't the case with axial diffusivity, and so in this case TBSS was like a lot less successful. Sorry, did that kind of?
Yeah. I mean, yeah, it kind of makes sense. It depends on. So what you're saying is they use different mechanism, and the region that you're detecting also affects the result. Anyways.
Yeah. So basically, like we're testing for differences in fractional anisotropy and FA. TBSS kind of works directly with the one-dimensional FA values for the entire time. So you can kind of bet that it's going to be there is kind of a greater likelihood that it's going to find kind of differences in magnitude between each one. I don't think, however, you're not really guaranteed. I think that those differences will be correct. You know, kind of, I think like in other studies of TBSS, it very often gives out false positives, and so they usually, and so it usually needs to be verified by other tests, but it's a good hypothesis maker. Sorry.
All right, thank you.
Okay.
Other questions?